So this is a big beginner's guide to RNA-seq analysis. So I'm going to go over some of the tools and the procedures that we use in our lab and what our experience has been over the few years. I'm not going to give a big in-depth comparison of different methods for doing this, but instead show you what we use and tell you about why we think these are good tools for this. And so just to remind you where this part fits into this overall schema of how we get from QTL analyses of complex traits that eventually identify a genomic region, and then how we can get to genes and eventually the biological pathway or mechanisms. And so this particular webinar is going to cover really this area in more of a broad sense of how we get from this genomic region to genes. And so RNA expression is one of our tools for doing that. And so what we're going to talk about today, or give you an outline, we'll talk about why RNA-seq, just a couple slides to that, a technical overview of the Illumina method for sequencing, which is one of the most popular technologies to do this, some of the considerations when it comes to making an experimental design. And then finally, we'll talk about how we get from RNA-seq reads to quantitative expression measurements, and then a common tool that's used for that differential expression analysis. So to start us off with why RNA-seq. RNA is one of my favorite things to study because it's one of the first quantitative links between DNA sequence and phenotype. So it's that first place for translation from what a variant that we may have identified in a QTL can, or possibly the pathway to affecting that behavioral or physiological phenotype. And so it really is that first step. And then from a statistics point of view or a systems genetics point of view, we also get real excited about the RNA dimension because we have all these fantastic tools, including graph theory, to really dig deeper into this data now that it's quantitative, to talk about interactions and possible hypothesized mechanisms to test. So that was why RNA. So why RNA-seq? So it's super popular now. There's not a lot of convincing people that it's valuable, but one of the things that we've had has had a profound impact on what we do is that not only can we do this quantitation of protein coding genes like we've been able to do in the past with a microarray, but now we can also do it for a variety of non-coding genes. So anything from microRNAs to long non-coding RNAs to SNOs to even circular RNAs, and how we can get more data for that full transcriptome, not just the items in the transcriptome that are translated into proteins. And we can also do discovery because we're realizing there's a lot we still don't know about the transcriptome. Despite some of the really in-depth work, we're discovering new things, a new role of RNA all the time. And so with RNA-seq, we're not relegated to only study things that we know are there, but we can also identify what else is there. So we can look at novel protein coding genes, novel splice variants, and novel non-coding transcripts that might not be in the database for your particular species or for really any species at all. And so that brings us to the technical overview. So if you're, this is meant to be a beginner's guide. So if you are a complete novice to RNA-seq, this is kind of the general overview of what happens. So you take a sample of interest, you isolate or extract the RNA. Then the next step is to decide what fraction of RNA you're most interested in and select that out of your pool of RNAs. Then it comes to the library prep step where we're taking those RNAs, we're breaking them up into small pieces, we're adding all the adapters to enable them to be sequenced on a sequencing machine. We're doing that sequencing here on, in this next step, we put them in a flow cell and put them through the machine. And what we get out are these reads that we eventually have to analyze and piece back together. The library prep step is that first step where, in this example, we've taken the poly part of it. We fragmented and primed it, and then we do a lot of synthesizing to get it down to this double-strand cDNA, so then we can add all our barcodes, and eventually it gets amplified on the system. The Illumina system for sequencing, so that includes your HiSeq, your NovaSeq, your MySeq, uses this technology where this is what we're describing when we're talking about a flow cell. And then you can see that within this flow cell, there should be 8 lanes for sequencing. And so what we're going to do is add our processed RNA that has been library prepped to these flow cells. And then this is what is going to get imaged and where we're going to get the reads sequenced from. So this is a really, as a statistician, this is a really fascinating process for me to understand. And I highly recommend this YouTube video. So it's a 5 to 6 minute video from the Illumina group that really, it's a cartoon that kind of takes you step by step through this whole process and talks about how these RNA pieces are tethered to the flow cell, and then how they go through this amplification process so that they get clusters of read or clusters of RNA transcripts that are identical, so that then we can use this high throughput sequencing. So they call it sequencing by synthesis, where as they add the matching base pair, they get this fluorescence, and then we read the color of the fluorescence to be related to the base that it's detecting. And so it starts at the beginning and just looks for different flashes of different colors. So that's a very broad overview from a non-technical expert. But the idea is, in the end, then we know this sequence of nucleotides that are matching our originally tethered piece of RNA or processed DNA. So one of the common things when people are first starting off into thinking about an RNA-seq project is the study design. And there's a lot more options than I think people first realize when thinking about studying up a RNA-seq experiment. And so the main elements that you need to make a decision on right away when talking to your core or talking to a for-profit business that would give you these read counts are which RNA fraction you're going to look at, and we'll go over that in a little bit more detail, the library type that we're going to do, the sequencing length, sequencing depth, and then number of replicates. So let's break these each one down individually. So RNA fraction, I tried to look for updated numbers. This is a little old, and they came from a few older publications. But when I look up this stuff, there's a lot of controversy on what exactly is being transcribed from the genome, how much of that is useful and biologically meaningful, and how that differs from tissue to tissue or sample to sample, depending on developmental stage, developing on environmental determinants, things like that. But in general, if we think of this light blue part of the square as being the full genome, then within this bigger circle are all the areas of the genome that can be, or we have some evidence of them being transcribed. The smaller part here is, I believe, the part that's being transcribed from both strands of the DNA. And when we look at protein coding, so coding sequences, they really make up a small portion of what's being transcribed from the DNA, not to mention the actual untranslated regions is what a UTR stands for. And then you also have some of these popular, smaller, non-coding transcripts that are coming from it. So the gist of this is that there's a lot of things that are being transcribed. And what we want to talk about, what we want to hypothesize about is going to drive which fraction we choose to take for our RNA-seq experiment. And we probably wouldn't want to take everything. And a lot of the reason why we wouldn't want to take everything is if we break down this total RNA distribution a bit more, we would get that only 5% of the total RNA is coding, meaning is translated into a protein, where about 80% is ribosomal RNA and about 15% is transfer RNA. And so there's a few different ways to decide on what fraction you want. So a typical traditional RNA-seq study, we're going to go in and pull anything out of our RNA pool that has a poly A tail. And so the reason why we're doing this is we're really trying to target those protein coding genes. Obviously, we're getting RNA transcripts that have a poly A tail, even if they're not protein coding, but that's the general idea between this poly A selection. We're also interested in the long non-codings. We can, instead of pulling out everything that has a poly A tail, what we can do instead is go into our RNA pool and take out or clear out some of that ribosomal RNA. And so what that does is it leaves room for us to measure everything else. Because if we didn't do this, our ribosomal RNA depletion, then 80% of our reads would align to our RNAs. So it's targeting both those messenger RNAs and our long non-coding RNAs. And then finally, the other tool that we can use in combination with, either one of these is a size selection. And so what a size selection does is say, I only want RNAs that are less than 30 base pairs. And what that will do will enrich our RNA for small RNAs and microRNAs that we can then run throughout our study. And the library type here, typically you have a choice between a paired end and a single end. For a lot of the studies, and I think especially the NovaSeq and a lot of the high throughput facilities now, they won't even give you the option of a single end read, that everybody just gets a paired end read. But the history behind it is that in the single read experiment, you're getting a read, whatever read length, and that's all you're getting, and it's good enough for every situation. In a paired end read, what we're doing is we're taking that piece of RNA and we're reading from both ends. And so what that does is that not only do we see how well this read 1 aligns to the genome, and we see how well read 2 aligns to the genome, but then we can also double check that alignment and see if it makes sense where these two were aligning, because they came from the same piece of RNA. So if the reads are aligning to 2 different chromosomes, then there may be something wrong with the alignment of that read, or it may help you choose, if the read aligns to multiple places, to attribute it to the area of the genome that makes these what we call concordant. And so sometimes in the beginning it could be more expensive, but like I said, now a lot of times we don't get an option and everything is paired. It gives us a little bit more power on getting unambiguous alignment of the reads, and especially when we come to do things like transcript discovery and isoform level expression, these types of reads are helpful. The other library type option that you'll often get is a stranded versus unstranded. And again, more often than not, we see stranded libraries nowadays, just because the pricing and the technology has come to make it that there's really not a whole lot of reason to do an unstranded. So in an unstranded read, during the reading of the sequences, we don't really pay attention to whether or not we're doing the complement DNA or the original RNA direction. And so if you have 2 transcripts like we do up here on the corner that overlap but are on different strands, we wouldn't be able to distinguish which reads belong to which gene. And so we'd have to deconvolute that later in the analysis steps. Whereas in our stranded libraries, we can tell precisely what's coming from which transcript, even though they overlap the genomic region. One of the next choices that you always get is sequencing length. So again, the longer you get, the more expensive you get. And some of the cores, some of the machines, you just get 150 and you don't get a choice. But in general, longer reads tend to improve mappability, improve accuracy of transcript identification. But when you're doing that small RNA fraction, definitely these longer reads are an overkill because you're really only interested in that 30 base pair region that you were targeting. The next question is sequencing depth. And this does vary from experiment to experiment. So usually when we say sequencing depth, what we mean is how many reads are you getting per sample or per library? And so really, the deeper you go, the more precise estimates you can get on more genes. And so this is what we're looking at if we're looking at, example, general transcriptome coverage. So the number of genes that have some kind of reads, we're going to get lines that look like this. The problem is that we don't know if we're on the purple line or the green line necessarily. So in general, the more reads, the more information you get, but at some point there is a diminishing returns. So one of the things I like to outline or recommend when you're thinking about sequencing depth, if what you're interested really is in a gene level analysis of protein coding genes, and you're looking at a relatively homogeneous tissue. So for example, a genome analysis with a known transcriptome, meaning that our databases like Ensemble and RefSeq have done a really good job of annotating genes for this particular species, then we can do a poly-A selected RNA in the liver for 20 to 30 million reads per sample and get pretty good coverage of most genes and have reasonable power for differential expression.
Laura, there's a question for you by Malad about the definition of strandedness, whether that refers to haplotypes or the plus or the minus strand in terms of 5' 3' UTR.
So it is respect to the sense or antisense strand, right? So it's which strand is that particular RNA coded from. So it doesn't give us necessarily haplotype structure. Does that help? Okay.
Perfect.
Thank you. All right, so we talked a little bit about kind of one of the most basic analyses where we can get away with maybe 20 million reads per sample. If what instead we're looking at is an isoform level analysis, where we're interested in discovering splice variants, we're interested in discovering non-coding RNAs, or we have a poor transcriptome to begin with, we usually need to go deeper. And what that means is if we want to do a really good job of defining our transcriptome, that often can mean 50 million reads, 70 million reads, something like that to get real deep. Obviously, most people aren't going to want to do it. And obviously, most experiments I see are really in that 20 to 30 range, unless you're doing a deep dive into it. And unfortunately, and we'll talk a little bit about this and how many samples to do in any kind of omics study that you do, and especially in RNA-Seq, a traditional sample size or power calculation is really hard because at a low sequencing depth, you're gonna have enough coverage for many genes. You may even have enough coverage for most genes, but you're not going to have enough coverage for all genes without doing millions and millions and millions of reads, right? And we don't go in knowing what is our gene of interest. So it's really hard, or what level of coverage our gene of interest will have. It's hard. It's very much a global power analysis. It's some of these global decisions. We're never going to catch everything, but our hope is to catch many things. So the quote from the paper is, "The number of reads that's required in an experiment is determined by the least abundant RNA species of interest." And so obviously we don't know what our species of our RNA species of interest is before doing the experiment. So the same thing is true about the number of replicates. Again, power is a little bit more difficult because not only do you have read coverage contributing to it, but you also have differences in within group variances and the desired detectable effect. I just wanted to show this table out of the Conessa paper to just kind of outline the few things that you can move around to really decide on the number of replicates per group that you need. Kind of a rule of thumb for inbred strains, which we do a lot of experiments on and we talk a lot about during these webinars. So really homogeneous groups, you can get away with, and you're doing a gene level analysis with 4 replicates per group, right? The more heterogeneous your groups are, or your treatments are, or your outcomes are, the more samples you're going to need. And again, you'll not have 80% power for all genes, but your hope is to design it in such a way as to have 80% power for most genes. Other things that can go into the sequencing part of it. So we've talked a little bit about the experimental design, and now we're actually coming to the part where we're making the libraries and putting them on the sequencer. So there are synthetic spike-ins available, and the goal of a synthetic spike-in is to give you an idea of technical effects, to give you an idea of, could we talk about absolute quantitation? And so these are transcripts that are not found in most species, and you can spike them in. They give you 2 mixes. This one always sounds great in theory. We, in our experience, have always put in external controls, these ERCCs, and have really struggled to get a significant amount of information from them. But I'm not giving up. I think that they're a good theory, and we're working with some different statistical methods on really how to take advantage of these to their full potential and really getting rid of some of our technical effects and understanding some of the issues that are coming up in the RNA-Seq. The biggest thing that I can preach about and will make the biggest effect is if you have enough samples that you have to do it in batches, to really be conscientious about making sure that you are randomizing at that library prep step. So there's lots of steps in doing an RNA-Seq experiment, but by far the library preparation step is the source of the most profound batch effects. So making sure that you are, if you have a two group comparison, that if you have 3 batches, you have 1 from each group in that, all the way through to the sequencing part. You can do randomization at the sequencing run, and that helps a lot. So with the Illumina, we can do now what we call multiplexing. So what that means is each sample gets a specific tag, and then we can pool all the samples and spread them across as many lanes as we want. That way, if a single lane fails, which I haven't ever seen in the whole time I've been doing RNA sequencing, that you would just lose a portion of your reads for all samples versus losing a few samples completely. So this technology has really helped a lot on getting rid of any kind of batch effects that may be due to lane or sequencing run, but we typically don't see a lot of variation in the sequencing part. So if we prep the same library and ran it on multiple days, or we ran it on 2 different lanes, we don't see many differences or batch effects there. So finally, getting to the transcriptome profiling part, so actually getting to the analysis. So thank you for bearing with me as we kind of set the stage for what we wanted to, the main topic of interest. And so how I'm going to break it down today is we're going to go over some of the things that we typically do to look at the quality of our raw reads and to make sure that we're getting good information out and we know what we're getting. we can do this alignment to the genome in a transcriptome discovery. So if you'll notice, these two bars are in green, which I am using to indicate that this is an optional step, that we don't necessarily need to do that. We can go straight from those raw reads to an alignment of a transcriptome and quantification if we're confident in our transcriptome. So we'll start off with this quality control of the raw read. So just to let you know, I am going to use this example data set throughout. It's available on GEO. It's a CRISPR-Cas9 knockout of LRAP. And my intent is to put some example code up with these lecture notes and this recording within the next week or so. And so it's 3 groups: wild type, heterozygotes, homozygotes. We've got 3 biological replicates per group. We did a whole brain sample. We did ribosomal RNA depletion of total RNA, and we're looking at paired end reads. And so if you've never seen a FASTQ file, whenever we do get a paired end reads, they come to us in 2 separate FASTQ files. So you can notice here that the names of the files are identical except for this R1 right here. So what this indicates is this is read 1 from each pair, and this is read 2 from each pair, and they're kept in what we call a FASTQ file. And then the .gz, if you're not familiar with it, just means that they're zipped, so that makes them smaller. A FASTQ file here below is an example of that. So each file has 4 rows per read. So this first row right here that starts with the @ sign is just giving us identification information for that read. And so, for example, these particular reads, sequencing was done at GeneWiz, and so that's their little symbol here, this GW, and then it was run on their HiSeq. The next line is the actual base pair calls, then a little bit of a spacer. And then these letters actually correspond to the quality of the call for each of these base pairs. And so we typically don't use it for alignment, but we can use this quality information to really check on the general quality of this read experiment. And we can even chop off some of these base pairs or these nucleotides as if they're of poor quality. And so in general, when we do quality at the base pair level, because of the nature of the technology, the quality of those base pair calls tends to drop off as the read gets longer. So the machine does a really good job at the beginning and slowly gets worse. But in general, they tend to look good. And so one of the things I always recommend is counting the number of raw reads and making sure that you have the equal number of reads between your 2 read sides for each read pair, making sure it's the length that you're expecting. And in general, we want to just compare across samples to make sure that all of them are between 20 million and 40 million, for instance. When we have a particular sample that is much different as far as library size goes from the other samples, it tends to create problems down the road. And so we just need to be made sure of that, or made aware of that earlier on, to be able to track that sample throughout the quality control process. In general, if you get low read counts for a sample, much lower than everyone else, then usually something went wrong with that sample. And so that's reason for concern. So if everybody else has 30 and you have 5 million for a single sample, that usually means that something's wrong, not that you should go ahead and use that 5 million reads, because they tend to look different than the other ones. And then finally, there are programs out there like FastQC that will look more in depth into the quality of these raw reads and looking at things like the drop off of the quality as the read gets longer. One of the things that we do in our lab is trimming. So we'll go into the reads and we'll chop off these adapters in case that the sequencer read through some of these adapters. We'll also chop off base pairs that are of low quality. In general, that's a little bit controversial. So this is a Williams, it's not Rob Williams, but it was a group that came out against trimming. The reason that we like trimming is because it is a good quality measure for us. So we can see if, for example, if RNA is degraded in a sample, what you'll see is that you're trimming a lot from those reads because the RNA went in too small. And so you're reading a lot of these adapter sequences. And so this is a really good point to double check that. And so we have seen samples that it looks like we have really good, a great number of reads. They look like all the other samples, but when we look at this trimming, they're having way more base pairs trimmed off it. They're having a lot of reads eliminated because after trimming, they're so small. But in general, when we do trimming, hardly any base pairs are trimmed off. Our average read length, if we start off with 100, is usually 99. And we get rid of very, very few reads for trimming process. So we're not as concerned about what it does downstream for our alignment as much as we are using it as a quality control measure. So if you're going through the path of transcript discovery, our next step is to align to the genome. So we're going to talk about aligning to the genome here first, because that's what you have to do for de novo transcriptome. But like we said, you can go right to the alignment to the transcriptome if you prefer. It's also come up in rodent populations like rats. If you have inbreds or you have heterozygous stock or heterogeneous stocks, excuse me, if you have their genotype information, you could align to a sample-specific genome. We haven't seen huge differences with this because a lot of our aligners are pretty robust to single nucleotide variants. So it'll still align there and just say that, oh, one of the base pairs were mismatched. But those are the options when it comes to the sample-specific versus reference genome. So this is one of the points where there's a lot of read alignment tools. We've looked at STAR is one that we've compared to HiSat2 before. The generation before HiSat2 before was Bowtie, if you've heard of that one. We found in HiSat2 to be a good balance of being fast and being accurate. And so what it does is it does a first couple of passes. And why we need these special aligners for RNA-Seq that are even more important than when we do something like DNA measurements is we have to be prepared for these gaps in alignment due to splice junctions, right? And we have to have specialized aligners that allow for these really large gaps from where the start of the read aligns to where the end of the read aligns. And it's really important that we don't just throw out those reads because they give us a ton of information about splicing in our data set. So we want to keep those in. This is just an example of the quality control that comes out when you align to the genome with something like HiSat2. So this is a simulated data set, so there's only 10,000 reads just to make it a little bit easier to look at and comprehend. But what it does is it tells us that, oh yes, you've paired all of your reads, and these are your primo sets. So these are your pairs that aligned concordantly, so meaning that the two sides of that paired end read align in reasonable places. And they only align once, but then we also get several that align multiple places and some that were not coordinate at all. So, but as we break these down, these are kind of our best case scenarios, but we do have a lot of single, just one of the pair aligns or one pair aligns multiple places and things like that. And so these general stats just break this down in much more detail and then give you an overall alignment rate at the end. And so the example here I have is about 96.7%, and that is generally what we see with a good data set with a good genome. So when we're talking about rat RN6, that's what we're seeing. When we do it to RN7, we get a little bit of boost. So there's a new version of the rat genome. We get a little bit of boost, like 1 or 2 percentage points, but we get a huge boost in this concordance rate. what you get out of that genome alignment is what we call a BAM file. So a BAM and SAM file contain the same information. BAM is a binary sequence one, so it takes up less space on your computer. So you typically wouldn't save anything in a SAM file because it would take up so much space. There's a whole suite of tools called SAM tools that are super easy to use that can do a lot of manipulation with these data files. We can sort them, we can filter them, we can flag them, we can get summary information from them using this set of SAM tools. We can merge two together. And in this data file, we get one row per alignment. So one read may have multiple alignments, so it will appear in multiple rows. Again, this is our unique identifier for that read. A flag is telling us how well it aligned. So it's giving us lots of qualities about the concordance and things like that. I have included on the GitHub for this particular lecture a website where you can plug in these numbers and it'll translate it to what all they mean. This is the chromosomic longitude, the position, the mapping quality, and then this cigar measurement, which is interesting in that it's 100, this particular read is 100 base pairs and all 100 base pairs match smoothly. So once we have it aligned to the genome, then we can do something for transcript discovery. And so when and why would you want to do a transcript discovery on your data set? So here, for example, I have looked at the latest version of Ensemble for 3 species, human, mouse, and rat. And so what you can see here is that when it comes to coding genes, they're pretty comparable across the 3 species, meaning we have about 20,000 coding genes. So we're feeling pretty confident that mouse and rat are well annotated at that gene level. When it comes to non-coding genes, we see much more of a discrepancy between the 3 species. And our general assumption here is that this is not due to evolution or something like that, that this is due to lack of annotation. And so, for example, there's only 3,000 non-coding genes that are annotated in rats, where there are almost 24,000 in human. So if we are targeting non-coding and are super interested in the non-coding transcripts in rat, then we definitely want to consider a transcript discovery type algorithm. And then this final column is one of the more drastic ones too. And this is, we've gone here from protein coding genes to gene transcripts. So this is splice variants per protein coding gene in addition to the non-coding genes. So what this is telling me is that in humans, we tend to have about 10 splice variants annotated for each gene. In rodents, we've got something like 7, where in rats, we have 1 or 2. So again, if you're interested in splicing, then transcript discovery is something that would be of interest to do. Whereas if you're only interested in doing a gene level quantitation, it may not be necessary to go through the transcript discovery phase. Again, there's lots of tools out there. We tend to use StringTie. It's got some pluses and minuses, but it's simple to use. There's some really good pipeline papers about how to use it effectively. But the general idea is that it looks at all these reads and builds what is called a splice graph and uses this algorithm then to determine how to put these exons together into a single transcript. And so that's both based on coverage of those exon junctions or splicing junctions, but also then the read depth at different places. Like I said, this is one of the good protocol papers that came out in Nature that really does a step-by-step guide on how to run through it to get this transcriptome. But in general, their recommendation is you build these sample-specific transcriptomes or do sample transcript discovery in an individual sample and then merge those assemblies later on. So there are some caveats to this whole process, but in our hands, we do see a significant improvement in alignment rates going from alignment to an ensemble transcriptome to a reconstructed or de novo transcriptome. And so this is just an example of what you get out of the transcript discovery is what's called a GTF formatted file. So it's basically a map of where all these transcripts are. And one of the best tools for visualizing the GTF is that you can simply upload it into the UC Santa Cruz genome browser. And so you can see here, these first 5 transcripts are ones that we derived from our transcriptome data. The REF-seq gene, this is one of our favorite genes, GNB1, is right here. So we can see that there's only one isoform that's annotated for it in our species, but we're finding 5. And if we look at non-rat REF-seq genes, we see lots of evidence that these are probably valid splice variants because they've been identified in other species. So this has been a good example of information that may not be in the database, but we have other sources that indicate that we're probably on the right track and it's worthwhile. I do want to point out a few of the caveats just because it's not perfect. So the accuracy of the reconstruction is still not optimal. We do get some false signals in there. We do get, it's not perfect. It's super terrible at the start and the stop sites, but that's really not what it was designed for. It was designed more for what we call exon chaining. But just to be aware that for instance, alternative polyadenylation is not identified using string tie. And finally, from a statistics point of view, our uncertainty in the structure is not propagated into that differential expression analysis. So when we take all of these transcripts on to do statistical tests in, we're not treating the ones that we're super confident in any different than the ones that were novel and maybe there. And so then the last part in this transcriptome profiling is alignment to the transcriptome and quantification. And so like we've talked about before, we can do this on the gene level analysis, or we can do it on the isoform level part. So gene level, we're super confident in, but because we don't have any of these ambiguous reads that align to multiple genes, or we have very few reads that align to multiple genes, but then you miss some of the information that may be important about different isoforms or splice variants having different functions, where the isoform level analysis is interesting, but there's more ambiguity on our actual read counts there. Again, one of our favorite methods for quantification is RSEM, which is RNA-Seq by expectation maximization. And so in this procedure, we use, we align the reads directly to the transcriptome, and those that align to multiple transcripts, we divvy it based on a probability model. And so back in 2016, one group compared several different of these quantitative measures, and RSEM in the real data set was near the top, wasn't first, but second, but very close. And in the simulation, well outperformed the other methods. But to give you a general idea of the concept behind RSEM, so this is a really oversimplified toy example of 2 isoforms that each have 3 exons. They share 2 of them perfectly, and the other 2 are different between the exons. This top row is the number of reads that align to each region. So in our first pass, we have 100 reads that align to this region. So we're just going to split them 50-50. So 50-50, but all 10 reads go to this one because it's exclusive to this isoform, and all 90 are exclusive to this one because that exon's exclusive. We add up the number of reads. So it's 190 to the first one, 110 And so we readjust our estimated ratio. And instead of splitting this 100 reads 50-50, we split it 63, 70, or 37 based on this 190 to 110 ratio. We do this whole exercise, calculate our number of reads per isoform again, and adjust our ratio once more. And we keep iterating in such a way until our numbers stabilize. And so when we do this RSEM, what we get out are estimated recounts, FPKM, which is fragments per kilobase of transcript per million mapped reads, and transcripts per million transcripts. And so this estimated read count is not adjusted for library size, which we'll talk a little bit about later. FPKM is adjusted both for the kilobases of the transcript in million mapped reads. Although it's popular, even the people who first coined it say do not use this. So there are definitely some biases in this FPKM, and they promote the transcripts per million transcripts above the FPKM. When we're talking about quality control of this quantification, we really want to do a lot of exploratory data analysis, principal component analysis, hierarchical clustering, negative log expression. And with all of these tools, what we're trying to do is really say, do these samples look similar? And what we're trying to do is identify an individual sample that may look like an outlier, maybe something that's had some problem along the process. The other key observation is, are we getting correlations? Are our samples similar that are from the same group than they are between groups, right? And one of the things that no one pointed out to us on the first process, but we've found extremely useful in some of our data sets, another metric beyond these simple exploratory data analysis and visualizations, is looking at the proportion of reads that align that are attributed to the top 10 transcripts, and the proportion of transcripts or genes within a sample that have a 0 read count. In this top 10 transcripts, a lot of times this is really interesting, especially in the context of the total RNA fraction, where you can get the majority of the reads aligning to something non-coding, or even to our RNA that wasn't washed away with the filtering process. And so that has been a good quality check for us. This is just an example, and I know I'm brushing over this. I'm way over time here, so some of these details you can feel free to read later, but I want to make sure we get through the differential expression. So here's just an example of a hierarchical clustering match and some of the things that we're particularly looking for. So as we move to the differential expression, so we've gotten our reads, we've designed our experiment, we've got them aligned, we now have that almighty, what we call a count matrix. So it's got genes or isoforms along the rows and samples across the columns, and we're ready to get into the nitty-gritty of our actual statistical hypothesis with this data set. But we do have to do some more pre-processing analyses to this. And one of the pre-processing steps that we often do is get rid of genes that have really low read counts. And the gist of all of this text is that we don't really know the most appropriate threshold. It depends on your threshold for noise and yourself. The best advice here is to make some kind of cutoff that you apply to everything that you feel comfortable in. And to get on my soapbox a little bit, my general philosophy is never perform a statistical test that you don't intend on interpreting. Meaning that if you got a result where a gene was differentially expressed between two groups and you came out really significant, but you only had 3 reads per sample in one group and none in the other, would you really spend your time and money following that up or interpreting that? And so thinking about what you yourself would need to feel like these results were biologically relevant. There's definitely a library size bias, so we never compare just raw reads to each other, right? So in this example, if we compared this first example to the second example, just looking at gene A, we'd say that there's twice as much expression for gene A in the second sample. In reality, it just had more reads. So RNA sequencing, just like microRNA or microarrays, is more of a relative abundance question than an absolute abundance question of these genes. So we not only have to talk about the read count for a gene, but how many reads we got for that entire sample. If you're doing a bigger study, so something that's going across batches, I guarantee there will be batch effects, and there'll be really strong batch effects. One method among many for getting rid of these, and it's slowly not becoming my favorite, is the remove unwanted variants, so the RUV method. And what that does is try to identify these latent factors in your data sets to then eliminate them from those latent factors from your actual statistical analysis. And so there's a package in R that's RUV-seq that you can identify these factors and then either adjust your read counts or put them directly into your differential expression model. The one thing that I did want to highlight before moving on too quickly is that in our hands, we have tried all three. There's 3 ways to identify the latent factors. You can use control samples or negative control genes. We've tried both of these because we have the spike in controls, and we do a technical replicate that's shared across all batches. But the empirically derived negative control genes tend to work much, much better than the other 2 methods for identifying these latent factors, even when they're present, the control samples or the negative control genes. So finally, getting you into the framework of what's the appropriate statistical model for differential expression, I wanted to show you these 2 samples across a couple different scenarios to kind of get your intuition and your instinct going. So if we look at a row as a gene, in this first row, it looks sample B has twice as many reads than sample A. And in the second row, sample B has twice as many reads than sample A. But instinctually, we know that the second gene is much more likely to be differentially expressed than the one that goes simply from 1 to 2. So we're much more confident in this difference than we are in this difference.
Laura, a question from David that I think we might want to handle right now. He asks, is RUV-seq or the equivalent still possible if you don't have a binary phenotype or binary categorization of samples?
So I think that's a good question. Hypothetically, it is still available. So you could put it in as, because some of the issue with some of these latent factor analyses is you don't want to adjust for your factor of interest, right? And so you need to tell the model, don't eliminate effects due to strain, don't eliminate effects due to genotype. So you would have to put that quantitative value into the RUV model. And the reason why it often works with those empirical negative controls is because you calculate those empirical, or you identify those empirical negative controls because they don't associate with your quantitative trait, right? And so as long as you're doing that to identify those empirical core control genes, it shouldn't matter what your phenotype of interest is. And so just getting back to the instincts here too, we also recognize that this 100 to 200 is a more dramatic change than 100 difference here between the 1100 and the 1200. And 0 to 2 is not very impressive, but 0 to 2000 probably is impressive. And so as we start thinking through these examples and as we start developing this instinct, we realize that some of our traditional like linear based tools aren't going to work for this data. And so what we need instead is what typically used for RNA-seq data is, instead of a linear model, we're using a negative binomial model. And so for a negative binomial model, the idea is how many failures do you have before a given number of successes? So in the context of, and I don't know if this is spurred from missing being in the office, but if our example is we want to find 5 people that have seen the movie Office Space, in the negative binomial model, we want to say how many people will we have to ask before we find those 5 people. And so just like our instincts from before, if we have to ask 20 people to invite 5 people, then we're guessing that about 25% of our population has seen Office Space. But if we instead switch it from 5 people to 50 people and our numbers get bigger, our confidence in the precision of our estimate gets bigger. And so when it comes to RNA-seq, we're going to use this type of model that deals with discrete observations, because we're talking about discrete reads, to identify genes that are expressed in different proportions across their groups. One of the most popular tools, again, not one of the only tools, but one of the ones that we've had some success with in our lab applying, is DE-seq2. So it uses this negative binomial model, but also uses this concept of shrinking, which helps stabilize the dispersion estimate. So a dispersion estimate in a negative binomial model is kind of like, conceptually wise, variance in a linear model. And so what we're doing here is using information from all our genes to say that our estimate of the dispersion for this particular gene is a little bit off. And so we're going to bring it down and make it a little bit better. And so this isn't anything new as far as this empirical Bayes method for handling dispersion. We've used it. If you were a microarray person, you probably used it in the Lima package. It's just a way to handle the smaller sample sizes and not being able to estimate really precisely that variance component of your model. It's easy to implement. It can handle multiple covariates. It even can compare between models. So we can do what's called an omnibus test. So if you think about a one-way ANOVA with 3 groups, you get that first test that says, is there any difference between all 3? And if they're any of the 3, and if they do, then you can break it down. We can do those types of omnibus tests in DE-seq2. One of the big drawbacks as RNA-seq becomes more accessible and we can do bigger studies is that it doesn't handle random effects. So we can't do longitudinal studies. We can't do replicates within a strain if strain is not our factor of interest, things like that. So the more complex statistical models we can't do with the current implementation of DE-seq2. But luckily, there are ways to transform our data to make it look somewhat normal so that we can use some of our other tools in our toolbox for statistics. False discovery rates, I'm not going to read all this because I'm over time, but in general, we're doing lots of comparisons. We've got to do some kind of multiple testing correction. And in most omics studies, we use something like a false discovery rate. And finally, there's some other tools. These are just resources for looking at alternative splicing and alternative polyadenylation. So jumping right to the conclusions, I'll let you read those on your own since we're out of time, and we'll probably have more fun talking about the discussion later. But I wanted to point out that this is sponsored by our center. We're trying to do some outreach here and do some training, and then finally grants and all the wonderful people in my lab and collaborators' labs and everything